Here all we'll learn about glucose biosensor. Glucose biosensors are usually used to check the blood glucose level at in the body. The samples used are usually blood, so that is why it is called as a blood glucose sensor. So what is this blood glucose means is the amount of glucose that is present in the blood. So after any metabolism, that is after you take a right quantity of the food, the unused glucose will remain in the blood. Over period of time, these glucose, if it is not being used as like all of the function of our organs like liver, brain, kidneys, muscle, glycogen, or any organs for that example requires the glucose for their functioning. So if you look at this picture, you can look at that brain requires 15 gram of glucose, liver for 17 gram, and for fat generation you require around 2. And for glycogen synthesis, glycogen is a stored form of energy in our body, so that requires 25 gram. Muscle mass increase is through 23 grams of glucose, and for kidney functioning you require 8 gram, that too in the form of lactate. So this is how our body functions. So all the organs in our body requires a quantity of glucose for its functioning. But in case if your blood glucose intake is more, that is your carbohydrate consumption is more, the blood glucose will keep increasing, and that will result in a condition called diabetes. So the blood glucose in the body should be regulated. So there are two ways how it is being regulated. So one is like uh, using insulin, the other one is through the glycogen. So what happens is that the beta cells of the pancreas will release the insulin, and this released insulin will regulate the blood glucose level in the body. In case of certain stimulus, what happens is that the blood glucose level will fall. So in such cases, the pancreas will form something called as, or release something called as glucagon. So this again goes to the liver, it breaks down the glycogen that is being stored in the liver and release glucose. So again, the blood glucose level increases, and the basic or the homeostasis is a condition where the blood glucose level is in balance. The basic level or the concentration of glucose that should be present is about 90 mg in 100 ml. Anything above this level is called as diabetes. So there are two pathways, insulin pathway, glucagon, to take care about the regulation of the blood glucose level. If your blood glucose level is more, it is called as a diabetic condition. So in the fasting level, your blood glucose should be around 80 to 100 mg per 100 ml, and after eating you should have around 170 to 200, and 2 to 3 hours of this is standard range or normal range. In case if you have an impaired glucose, then you have the levels as such, which is a orange signal or a yellow signal for diabetes. So if it is called, this condition is actually called as a pre-diabetic condition. So that is impaired glucose. If it is present, the levels are slightly higher. You don't call it as a diabetes, but yes, there are always chances if the person is not managing his glucose level, he may go into the diabetic condition. So diabetic condition is around like 126 mg and more, and after eating it goes till 300, and 2 to 3 hours more than that it goes to 200 plus. So to analyze the glucose level in the blood, you require an analytical device. And if you look at the Indian statistics, that more than 70% of the people in India are diabetic. So you require a basic sensor to check the glucose level, or else what happens is that you cannot regulate or you cannot metabolize the correct amount of glucose or keep your body in proper condition. So now when you look at the glucose biosensors, it's an analytical device that is used for the detection of the glucose in the bloodstream. And how easy you can detect, so much will be the popularity of the biosensor. So as you are aware, all of them, all the diabetic persons nowadays, they do have a glucometer at their home to check the blood glucose level. They need not go to a clinic, give their blood samples before and after. They can comfortably be at home and check the blood glucose level. So coming back to how this glucose has been glucose sensor or the glucose sensor is being developed is in three generations. So the first generation, second generation, and third generation, and they are basically based upon enzymatic biosensor. The enzyme used in the sensor are called as glucose oxidase. So these enzymes will take care of the sensing mechanism. So in the first generation glucose sensor, so this is to tell you like what advancement has happened over the years and how the still more researches are going on, especially on the glucose sensor. As I told you, more people in India, that is more than 70% of the population, are diabetic, and there is a requirement of checking the glucose during the stress level, during a normal functioning day or less exercise day. It's a continuous monitoring that has to be done. But now let us see like how these glucose sensors are evolved over period of time. So the first design is called as first generation glucose sensor, and that was developed in the year of 1953. So here what happens is that they used a enzyme called as glucose oxidase, and this enzyme when it comes in contact with the glucose, it will release gluconic acid. So the gluconic acid that is being generated by this process will be detected by an electrode. So the electrode what they used is normal Clark's oxygen electrode. So this glucose oxidase will be immobilized in a glass permeable membrane, and this will consist of a platinum cathode and a silver anode, which will enable the detection. So looking at the diagram, this is a reaction. Here glucose, when it comes in contact with the glucose oxidase, gluconic acid is produced. So as a gluconic acid is being produced, it's an acid, so your pH will drop down. So there will be a drop in pH. So the ordinary pH electrode can be used for the detection of this change in pH. So here is an electrode. Okay, so as I told you, the platinum is the cathode here, and the anode that is being present is a Ag, that is silver anode. So there will be an electrolyte, and there will be a, usually if you look at the pH probe, the glass at the bottom will be a semi-permeable glass membrane. Similarly, here we use a semi-permeable glass membrane, and in that we have immobilized the glucose oxidase enzyme. So when it comes in contact with the sample, what happens is that the glucose oxidase will react with the glucose, and gluconic acid will be produced. So the production of this gluconic acid will be proportional to the amount of glucose that is being present. So now when this acid is being produced, there is a movement of ions to the cathode and the anode due to the change in H plus ion. So as there is a change, there will be a deflection and the signal can be monitored here. So that was the first type of biosensor that is being developed in the glucose. So there were some drawbacks for it. One thing is like if the high amount of operating potential is being used, the selectivity became a question. So when the potential difference is less or too much more, how selective your enzyme will be or how selective this signal transduction will happen, there was a problem in that. And there was a problem with the solubility of the oxygen in the biological fluids. So that is when the some of the biological fluids were used, the density of the liquid will be more, and which cannot exactly locate how much of glucose was there. And deactivation of the enzyme due to the production of hydrogen peroxide. So the enzymes are very sensitive, as you know, and they are protein substance. So when the hydrogen peroxide was formed, there was a deactivation of them. So because of this drawbacks, they went into the second generation glucose sensor, which happened in almost in the year of 1970s. So here, instead of using the oxygen electrodes, because as I told in the previous thing, so when some biological fluids, especially blood, is being used here, so the viscosity and the density of the blood will be more, so the solubility of the oxygen will be again less. So because of that, there was variation in the recording or the signal production. So this drawback is being solved in the second generation, where they try to replace this uh, oxygen electrode with the electron transfer agents. So these electron transfer agents are called as mediators. So there were a lot of mediators they started using: ferrocyanine, ferricyanine, quinines, methylene blue. So all this were used as a mediators instead of using the oxygen electrode there. And these mediator helped in the electron transfer, and the accuracy becomes still more better. So now here, yes, this is a glucose oxidase enzyme that is being used. So instead of using a oxygen electrode, what is being used is like electrodes are used, but still there is a mediator, because in the first generation sensor, the major drawback was the solubility of the oxygen. So if you use a mediator there, that is ferrocyanine or ferrocene or ferricyanide, quinines. So if you use a mediator here, the solubility of the oxygen will still become better, and the electrode can detect the signal very well. So that was the idea here, and it was replaced by the mediator. Yes, the accuracy and the sensitivity of the sensor still became better, but again this had some drawbacks. So what are the drawbacks of is that there was a lot of competition between the redox mediator and the oxygen. So the competition between them increased, which resulted in some inaccurate result. So if you're using a very minute amount of the sample, what happens is that the mediators will compete for the oxygen content there, and that became a problem, or the region between the enzyme and the electrode, there was a false passage of the redox ions which was happening, which led to the wrong readings again. So the third generation glucose sensor was developed. Here there is no mediator, but there is always a usage of a direct electron transfer. So here, in the diagram itself, you can see the electrode is placed, the enzyme is placed, but there is no mediator, there is a direct electron transfer. So how did this electron transfer directly happen? When the solubility of the oxygen became a problem before, in the first generation glucose sensor. So they tried to use some redox cofactor like FAH and FADH2 molecules. So this increased the electron transfer, and they made the better solubility of the oxygen too. So the molecules like tetracyanoquinomethane, which is usually called as TCNQ, and tetrathiafulvalene, which is called as TTF, are incorporated on the surface. So these molecules did the direct electron transfer instead of a mediator. Even for our works, when we have a mediator to do a work, if the mediator doesn't perform well, the work will not be accomplished, or there may be a false projection of the actual work by the mediator. So always it is better to deviate or to remove a mediator in a process. So similarly here also, FAD and FADH2 molecules were used as instead of mediators, which facilitate the direct electron transfer of the molecule. So these are highly reversible and stable for many enzymes, and this kind of things helped in the better electrode formation. And the electrodes also, they tried making with finite powder of graphite and fine crystals, which helped in the still manufacturing. So here now, if you look at the first generation, second generation, and third generation sensor development, the first generation, what happened is that they used an oxygen electrode. So here in the second generation and the oxygen electrode, what the problem is was when you use a blood, the solubility of the oxygen became a problem. So the glucose, the reaction is that the glucose oxidase enzyme will be used as a sensing part. And now when the glucose turn into gluconic acid, pH changes. But if the solubility of the oxygen is not good, the probe gave a very wrong reading. That is the electron transfer. There will be a problem because of the less solubility of the oxygen and less redox potential. Because of that, there were false readings in that. So in the second generation, what happened is they started using a mediator. So the mediator is nothing but like ferricyanide, just to improve the solubility of the oxygen. Even in the second generation, there were issues. So there were false positives and false negatives, and the redox reaction became a problem. In the third generation, what happened is that there was a direct electron transfer without a cofactor, which enabled a more accurate readings. So there are like non-enzymatic glucose biosensors too. The reason is that when you are using a enzymatic glucose sensor, the enzymes undergo certain change in their receptors, or they may not be sometimes active due to various change in pH and other ionic transfer. So to avoid that problems, they are even now come up with the non-enzymatic glucose biosensors, where they use only a kind of receptor molecules, which is called as a fourth generation. So here, not even the enzyme will be used, only a similar kind of chemically synthesized receptor, which is similar to that enzyme substrate reaction, will be used. So additionally, they have even imbibed carbon nanotube to this electrode. So if you are using a carbon nanotube, the selectiveness of the detection of the glucose or the sensitivity of the sensors will be improved. So they use this glucose oxidase, which are like immobilized or artificially synthesized, and only the amine residues will be actually present in the form of receptors. And when there is a contact between these receptors and the enzyme, there will be a proper transfer of the electrons there, which almost avoided all the other problems. So this is the recently developed or in development using a carbon nanotubes, which improves the selectivity. And these are the protein residues or the amine residues which resembles the enzymes, where the contact point is glucose. When it comes in contact with that, gluconic acid will be produced, and this acid can be easily detected through the electron transfer through a probe. So this was developed again, and then more selectivity will be increased by using. So whenever you're using a nanoparticles, just remember that the selectivity and the sensitivity of the uh, sensor will go high than using electrodes here. So the carbon nanotube is used, and without the enzyme, there is kind of amine residue that is being used, and that can be detected easily. This is our fourth generation biosensors of a glucose, where actual enzyme is not being used and actual electrode is not being used. The enzyme is being replaced by the amine residues, and which will resemble the enzyme part, or even if when the enzyme is used, they are covalently and immobilized and attached to a matrix, so that they don't change their properties even when there is a change in pH or change in ionic transfer, etc. And use of carbon nanotubes or the nanoparticles for the electrode formation has made the sensor in smaller size more effective, more sensitive. Hope you understood today's class. And that's all for today. And further, we will be having case studies with respect to these sensors. So, what happens when a nanoparticle is impied, how the sensitivity is changing, how the selectivity is changing. Further, with more case examples, we'll meet in the further class. Thanks for listening. Thanks for your time. I'll end my class here. Thank you all.